Yeah, so I'll briefly introduce to the interests of a group, and then I'll talk about some previous work before I'll dive into this recently published project on more on the evolution side. But we are in the group, we're very interested in understanding the cellular consequence of genetic variation. So that means we would like to understand and model how changes that happen in DNA end up causing some change at the level of organism fitness. And so for these reasons, we've also often worked with structural protein structures. So structures are for us to understand, well, first of all, to understand the function of the protein itself. They're very important to understand the rationalization or rationalize the impact of a mutation. So for example, we would like to be able to say if a mutation impacts on stability or interactions and so on. And I'll briefly mention some of this. And it is also important for studying evolution. That's going to be really the focus of the presentation here today. And so we've been working with structural biology now for a long time, but then, of course, as with many others, the appearance of AlphaFold really changed a lot how we could use protein structures, since in principle we can use structures from sequence now. And so together with many other scientists that work on structural bioinformatics, we quickly after the AlphaFold 2 was released, we tried to apply it to a number of different applications. And by and large, from the things that we could try that on, it seemed that these predicted structures were as good as experimental models if we took care of the uncertainty. That just means, as you probably all are very familiar with, AlphaFold comes also with very good uncertainty measurements. And so if you filter by these uncertainties, or if you take that uncertainty score into your downstream application, then these models are extremely good. And so, oh, sorry about that. There's some slides here with some blue background, but just a few. So apologies for the background. So what we're trying to use these models for now is to try to understand the mechanisms of action of these variants. And the variant impact predictors that are derived from sequence models, such as alignment-based models or protein language models, they are very good at predicting when the variant is likely to be pathogenic. But they are not telling you how that pathogenicity happens. So they doesn't tell you how that mutation causes an impact on stability or interactions and so on. And this is where the structures are very good for. This protein structure information is better suited to tell you about the mechanisms of action of the variant, and they're less accurate at predicting pathogenicity. And so one of the things that we tried to do together with our Alphason group was to predict structures of protein complexes. And in this case, we initially tried this on a set of about 65,000 human protein interactions. And we could see that these predicted models were very good as well. So on average, we use either known PDB structures, or we used an external data, which is cross-linking MS, that tells us where two positions in a protein sequence can be connected by a certain distance constraint based on the proteomic data, which is these blue lines here. So they should be less than 13 angstroms away in space if your model is correct. So based on information, we could say, based on all of the predictions for which there is some cross-linking data, about half of our models were correct based on this definition of correct. And we could also take internal confidence estimates, and I'm happy to talk about these confidence estimates, where we could be more stringent, and then we get more models being correct, but then we lose quite a large number of these models. And so once we get these models, we can then overlap mutational data, and we can say whether some mutations would be at the interface. There are some computational tools also to try to predict when a mutation has a specific effect on binding affinity as well. We didn't do so much into that, but there's a possibility. So just to say where we are at the moment, we've expanded this now quite a lot. So we've now made predictions for 500,000 protein pairs. And there's a lot that we derive from this, which I'm not going to go into. So really the biggest issue that we have at the moment is it's computationally quite intensive. So if you wanted to do all protein interactions, it'd still be quite computationally intensive to run. And it's also very hard for us to prioritize which protein interactions to model first. And the problem is about 1 in 5 of these we think are high confidence models. So 4 out of 5, we waste a prediction, in the sense that we waste compute effort and we don't have a reliable model in the end. So the other thing that we looked at is using structures to try to identify pockets. And of course, pockets are very interesting because of small molecule binding and drug discovery. And very early on, when we started looking into the AlphaFold predictions, one thing that's very clear is that sometimes you can have artificial pockets being generated through the fact that AlphaFold will still try to create a structure for the whole protein, even if it's uncertain about the relative positioning. So here's an example that's very easy to illustrate. So this is the EGFR receptor. This is a kinase receptor that's at the membrane. And the right side of this, you can see what the structure should in principle look like. There's a transmembrane region. There's an intercellular region and an extracellular region. And the extracellular region should not be in contact with the intercellular region, but AlphaFold does not know what the membrane is, and it does not separate this. In fact, it predicts a contact between this extracellular region and this intercellular and transmembrane region down below. And here in the middle, there is a really highly predicted, well-confident predicted pocket, which of course is completely artificial. And so Jürgen had been trying to, whoops, sorry again for these blue backgrounds. But so what Jürgen was trying to do is come up with a score, which is here at the bottom. This is unpublished work, where along with these scores that are often determined, such as the type of pocket, the properties of the pocket, Jürgen also included the information about the uncertainty of the pocket residues as estimated by AlphaFold. And so when we put these things together, we get a slight improvement in the prediction accuracy when we try to predict non-binding sites. So here at the top, we have non-binding sites from crystal structures, and we're ranking these according to this score. At the top, we have these. Oftentimes, we are better at predicting these non-binding sites compared to random expectations, so much better than random. So these are all tending to be here at the top. Of course, this could be biased because non-binding sites are taken from the crystal structures, and crystal structures are in the training set. So the other thing that Jürgen tested was, if I take a series of structures for proteins that have catalytic activity, so these are enzymes for which no homology model was possible to make. So we couldn't make this, we couldn't come up with a structure even by homology models. So then if we rely only on AlphaFold predictions, which should not be biased by the training data, are we able to predict back these catalytic enzyme proteins? And the rationale is, if they have a pocket, then they should more likely be an enzyme. And they should be ranking towards the top of this. And this is what we see. So we can predict enzymes purely based on the fact that we predict pockets in these. And so based on a not very stringent cutoff, we find about 100,000 pockets in the human proteome. And the majority of these pockets could not have been predicted by a homology model. So in green would be the fraction of the pockets that could be fully predicted by experimental or homology models. The red would be cases where none of the residues are even in homology models, and then orange is somewhere in between, where there's at least one residue that does not fall within an homology model. And so then we can also see that these predicted pockets tend to be enriched in disease variants, which is expected as well, but it's nice to see. So this is ranking pockets by this estimated confidence score, with a 4 being the top most stringent pocket prediction scores on a percentile or quantile. And these tend to be, so here's the odd ratios from the Fisher exact test. So these are much more likely to be enriched in disease variants. And we find pockets where the disease variants are specifically enriched in the pocket itself. And that's, of course, interesting because it suggests that the disease or the problem is more likely to be a problem of actually perturbing the pocket instead of perturbing the protein. There would be many other ways that I could perturb the function of the protein, not specifically perturbing the function of the pocket. So this is what we're trying to establish now. So we're trying to establish a procedure where we can use, we can take in variants from patients, and we could classify them on whether they're pathogenic or benign using sequence-based methods. So here we're using E, but there are other ones such as alpha missense or ESM. And so these are very good at calling out which ones are pathogenic. And then we use a protein structure to come up with a mechanism. So whether these mutations are destabilizing, they are impacting on protein-protein interactions, or they're impacting on pockets. And so we hope this For diagnostic purposes, what we really want is this middle part. But then for understanding the mechanisms, what we want is this structural aspect here. Okay, so that was just some example of some unpublished work, which I'm happy to explain in more detail if you need. And so then I'm just going to explain this recently published work where we looked at the structures that are available in the AlphaFold database. And so there's over 200 million predicted structures from AlphaFold that have been deposited, and we really needed some way of trying to understand what is this universe of proteins that exists here. So in order to do that, the obvious question is we would like to be able to cluster them to identify groups of similar structures. And this is important because protein structures tend to remain identical for longer than the sequence. Here's just an example where you have two structures for kinases. One is a protein kinase. So these are two non-protein kinases. They have quite a very similar structure, but they're 20% sequence identical. And this happens quite a lot, where the structure stays similar over evolutionary periods of time longer than the sequence. However, if you wanted to compare structures, although there are fast methods, they are still not scalable to millions of comparisons, or even hundreds of millions of comparisons. So this is where work by Martin Steinicker's lab comes in, and he's developed a really nice approach to encode protein structure information as an alphabet. And so the way he has done this is they train a variation autoencoder to take in positions of proteins in three-dimensional space and encode those into an alphabet, where instead of having a residue such as an amino acid, alanine, lysine, etc., this is encoded into a three-dimensional alphabet that understands, or in principle understands, the structural environment of that position. And so in this way, you can take a sequence, a structure, and encode it into a sequence. And that sequence is now a representation of that structure. And so this allows Martin, allows anyone now to compare protein structures by comparing these vectors, these sequences, because we have many, many very fast tools for comparison of sequences. And so he implemented this in FoldSeq. This is available for anybody to search a particular structure against a database. It works a little bit like BLAST. And so in this more recent study that we did together, so it was done by Inigo in my lab. Yinji is a student in Martin's lab. And so Martin and myself were co-leading this with other collaborators. So the additional trick that was added beyond FoldSeq was that this was implemented in the linked list algorithm. And so essentially, technically, the idea is we don't need to do an all-by-all comparison, but essentially, so new sequences, new structures are added to pre-existing clusters. And so this way, the clustering can grow in a linear sense. And that allows us to do this very large clustering of protein structures. So this is, in the end, the whole procedure. So initially, the 200 million proteins were initially clustered by sequence similarity. And so this is taking proteins that are sequence identical with 50 or more % sequence identity over 90% of the overlap of the sequences. So this is about 50 million clusters. From each of these 50 million clusters, the highest PLDTT were selected as a representative. And then these were again clustered now using the FoldSeq clustering algorithm, and in this linked list implementation that allows us to do this in a very highly scalable fashion. So then this leads to about 2.2 million clusters, which are non-singleton. These are more than one, and about 13 million singleton clusters. So we can say there's about, or in the order of 2 million out of the 200 million, there's about 2 million non-redundant clusters. And then there's many singletons where they just look like one example. So there could be a lot of errors here, or they simply just don't look like anything else. So one of the questions is how much of this structural space is new, because that's of course a question that people would ask is, how much of these represent new folds or new families that were never seen before. In order to do that, we compare this against the PDB. We compare this against known domain family sequence models, such as Interpro, those stored in Interpro. And we looked at any sort of domain and family annotations for any member within the cluster. This is quite restrictive. So to give an example, if you had a partial blast hit to the PDB sequence within the whole protein, that would already signal as this has been seen before. But in fact, it could be that it only represents 10% of the protein structure or sequence. And therefore, there could be a lot of it that's not yet seen before, but then we mark this as something that's been seen before. So when we do this, and again, I think this is a bit stringent, we get about 30% of the clusters without any hits on any of these types of annotations. So 30% of the clusters represent new types of proteins. But these only represent 5% of the protein IDs. And the reason why this only represents 5% is that the bigger size the cluster is, the more likely is it that we would find an annotation. So this makes sense both statistically speaking and also biologically speaking. So big clusters, so here's the cluster size. If we have very big clusters with many representatives, then these are likely to have some form of annotation. So the rare clusters that have very few representatives, then those are more likely to have no annotations. And so in the end, you could see it both ways. There's probably more than 30%, but at least 30% of the folds, let's say, if you want to call it that way, are new, but then they really only represent about 5% of the actual protein sequence space. So most of the protein sequence space that's in the AlphaFold DB is in some way matching things that we already know before. This is, of course, not counting the singletons, but the singletons would then represent also a relatively small proportion of the sequences. Okay, so then what we looked at is within this new things, could we try to predict some functions within these new things? And the thing that we use DeepFry, which is a neural network model that uses protein structure to predict function. And we just, in this case, we took 47,000 non-annotated clusters where the representative have a really high prediction score from AlphaFold. From these, we also predicted pockets. And then among these, we asked what type of functions does DeepFry predict? And at the very top of these, most of these were predicted as transporters, membrane proteins and membrane transporters. And this is perhaps not unexpected. So a lot of the structural space that has not yet been studied is membrane-bound proteins. And that probably has to do with the fact that membrane-bound proteins are very difficult to study using structural approaches. So here's an example, but there are many such examples of predictions where there's this really diversity of membrane proteins that has been predicted, and for which there is no structural characterization as either domain family annotations or matches to PDB. And then we also found many interesting examples that are worth then maybe in the future pursuing. One example here is an example of something that looks like a structural maintenance of chromosome type family. Here in the middle, so in the right, we have a known family member, RAC-N. And so these are forming these types of structures where you have a dimer, and so part of this would bind DNA. And there are some very specific protein motifs that usually are found in this family. And so here on the middle, you have this green protein that looks vaguely like the one on the left, but really doesn't look that similar. And so, but DeepFry predicts this as maybe having these types of functions, but it's sort of missing here in the middle some of these motifs that could be the. However, this particular protein sits next to our RAC-N protein in the genome of this species. And so it makes sense because in bacteria, proteins that sit in the genome together tend to be functionally connected. So it could be that this particular protein actually works in some form in DNA maintenance related functions. So one example that I really like is this something that vaguely resembles a kinase. So here on the right, we have a protein kinase. We study a lot kinase signaling, therefore This is very recognizable for me. So here on the left side, we have something that, structurally speaking, is very different. So there are a lot of structural aspects of this are very different. But on the other hand, there's many things that look similar. So here in the middle, we have something that could resemble an ATP binding pocket, and we have this upper lobe and this lower lobe, something where you could imagine that could be a target, sort of a binding pocket for the target here in the middle. But then there's sort of weird things like this weird helix bending here towards the left that doesn't look super plausible. When we were doing this, we found many things that look like errors. So we found examples of proteins with very high PLDTT. So these are all cases that have a higher than average 90 PLDTT. So AlphaFold thinks this is a completely excellent prediction. But then 80% of the protein is accessible to the outside, forming something that sort of is artificially predicted as a pocket. And there are a few of these examples, not many, but there are several of these examples. And this does not look like a well-folded protein, of course, not in any way, but AlphaFold is super confident about this prediction. And so this is, interesting cases of things that are for sure, most likely errors from the model. So finally, one thing that we wanted to look at is the evolution of these clusters, or how old are these 2 million different types of protein structures. And this is really not sort of the most accurate way of doing this, but what we did is we simply take the taxonomic tree, and then we say, if I take a given cluster, and let's imagine that cluster had 3 representatives, and the 3 representatives were found in these 3 species. So then what we could suggest is that the origin of this cluster could have happened at the base of that is shared between these 3 species. And this is not really a sort of evolutionary model in any sense. So we don't consider the rate of potential change, so gain and losses of proteins and so on. This is really just giving a plausible lowest possible origin for this type of cluster. And there are many limitations of this. Of course, the issue is we're talking about 2 million clusters, each of these clusters having potentially many representatives. And so it is computationally not very feasible to do proper evolutionary analysis across these 2 million structures, 2 million clusters. So taking into account this limitation, so here's where we look at all these 2 million clusters, and then we ask, where are they found across the tree of life? And so what we see is that the majority of these are found in very ancient form. So basically, they're either seen in the ancestor of all life, about 20% of these. About 60% are seen only in bacteria, but in the ancestor of bacteria. And about 30% in the ancestors of eukaryotic species. So the sum of this means that the majority are quite ancient. The surprising finding was that about 4% of these appear to be specific to a given species. And again, this is unexpected because, as I said, protein structures tend to remain similar for a long time. And it's unexpected you'd have the emergence of a new fold within the time frame of one species. And this could be that you could have de novo gene birth events that could lead to the emergence of new genes. But most likely, this should be also highly enriched in errors. But if they're not errors, then these are super exciting examples of potential novel gene birth events. What this allows us to do is then we can take, for example, if we take the human proteins, which is, and we ask what's the gene ontology that they belong to. We can look, for example, of proteins that have to do with very basic cell chemistry, such as ligase activity, for example, or peptidases. And we can ask the proteins of this group, where do they tend to originate from in terms of evolution? And so then this is what we show here with this heat map plot. So most human proteins that have a ligase activity belong to clusters that are predicted to have been originated in the origin of all life. So if we do this, then we can sort of have this evolutionary description for the origin of different functions for human cells. So as you expect, for example, things that have to do with immunity or hormones. So these are things that are not found in bacteria or not expected to be found in bacteria. And then we see that, in fact, these things are predicted to have been emerging in chordates, for example. However, within these, we found examples where, on average, they tend to be more recently evolved systems, but we find examples of them in bacteria. So this is a well-known example for histones. This is something that for a long time, is thought to be eukaryotic specific, that only eukaryotic species had histones. But then people have found that there are structurally similar things in bacteria. And in fact, that these probably have a common origin. And so we found this as well. But we found many other examples. And so just an example, a couple of examples of this. So here we have an immune-related protein. And in this case, the function of this protein is known in human. This is called bactericidal permeability increasing protein. So this is meant to be helping in fighting bacteria. And here on the right side, we found bacterial cases that would not have been predicted easily by sequence. And we also find partial matches. So if you look here, this is not a complete match to the one on the left, but it's sort of, if you split this in half, you see that this part on the right matches very well, reasonably well, this partial match. And this is interesting because this protein in bacteria, for example, in E. coli, it does not have a known function. So this does not have an annotated function in bacteria, but it's structurally similar to these proteins in human that are well characterized. And so this suggests that this protein in E. coli is capable of binding lipids and maybe involved in the lipid biosynthesis or maintenance. So just another example here on the left side, we have a well-characterized human protein that again is part of the innate immune system. It's part of this interferon-inducible protein, and this is important for, again, immune response in human. And we find examples of structurally similar proteins in bacteria. And so this suggests that this particular immune system has been adapted from things that pre-existed sort of mammals for a very long time. So finally, I'm just going to give you one more type of analysis that we've done in this study. So all that I showed you up until this point was when we were comparing the full length protein. So all the matches were done at 90% length similarity. So another thing that we were asked ourselves is whether we could use these structural matches to predict domain families directly. So not the full length, but potentially partial matches. So we developed a very simple method, but that seems to be working quite efficiently, is that if we take a protein, like this protein shown here in the bottom, and then we structurally compare it with many other proteins, we would find many of these local hits through fold-seq. And so what we ask ourselves is, if we take these local hits, these start and end positions, and we cluster these, we could in principle directly predict the start and end positions of these domain regions. And by having these clustering results, we could in principle decide that this region is structurally similar to all these other regions here, and together they form a domain family. And so this is what we've done. So then we have, instead of here we having proteins, we have domain families together. Or in this case, these would be individual domain sequences. And if we cluster these, we find these domain families. And so then we ask if this particular region of the clustering overlaps any PFAM annotations. And what we discovered, this is just a schematic, but we find these cases where we find that, for example, one of these clusters is primarily annotated to one particular PFAM domain. But then we have other cases where this other region, this other cluster annotates to another PFAM domain. But there is some connection between these. So in fact, there is some structural similarity between this PFAM1 and this PFAM2. And of course, then we have examples where there is another group here that has no PFAM annotations. So altogether, in this particular analysis, we found about 10,000 new families. Well, potentially new. I'm going to show you how many of these are new or not. But then this is limited by the number of starting proteins. And we only started with the proteins that were representatives from the 2 million clusters. And so there's potentially a lot more that could be discovered this way. So here's an example where we did this. So this is 3 clusters derived based on this type of analysis that are connected together. And some of these, the ones here in red, had annotations for a PFAM domain called the Gauss-Derman domain. And these other 2 did not have such annotations. So these are structurally similar to the PFAM domain called Gauss-Derman, but there are these other 2 clusters that are structurally similar, but don't have this PFAM annotation. And this was interesting because there had, at the same time, more or less, a bit before, there was this publication where they showed that, in fact, they discovered a bacterial form of Gauss-Derman. And so we essentially made the rediscovery of the same thing. In this paper, they've solved the structure. In our case, we just used AlphaFold. But there were, just suggesting that this approach really works and that there's potentially many other such examples in our data. And one final example, so this is the anthrax, which is very famous for the anthrax toxin. And here again, we have in red, we have a part of this clustering that's annotated by PFAM to be one of these anthrax toxins. And we find another cluster of these that does not match the PFAM annotations. And if you look at these, they are not a complete match, but they are a partial match between the red part here, which annotates to PFAM, and the blue one, which does not annotate to PFAM. Okay, so I'll summarize here, and I'll let you ask questions if you have any. So we took these 200 million structures, and using the criteria that we used here, we clustered the 2 million. Of course, there are many ways of defining the thresholds. So again, we used a 90% length, and there's always ways of changing the clustering to get more or fewer clusters. And of course, we haven't done that purely because computationally it would be quite difficult. Out of these 2 million, 30% may be new, but then this matches less than 10%, probably less than 5% of the protein IDs. But we think this is an underestimation. There's probably more things that are not known yet within this group. There are, I think, many novel protein families to study, many new enzymes, and certainly many new transporters. And this is for biotechnological purposes. This could be interesting, the identification of new enzymes with new forms of catalysis. There's many that seem species specific, and I would have to imagine that most of these are errors. But if these are not errors, then this is interesting because it suggests that there could be a fairly rapid de novo gene birth events happening that look like something that's structured. We find many examples of this remote homology, which led to some rethinking of the evolution of some of these systems. And we show that local hits can be used to find these domain families. So what's the outlook? So of course, that was 200 million protein structures that were in the AlphaFold DB. But in fact, there are over 3 billion sequences. And of course, there is already another database, the SM-Fold database, where there's a lot of metagenomics related protein sequences that have been predicted. And so that's an obvious direction to go into. We need better models that look at evolution by combining protein structure. And so recently, Christoph Desimov's lab have actually put out a preprint that shows that this is possible to do evolutionary models that include structural information. And then we find this, we can connect protein sequences by structural similarity across the protein universe. And I find this idea very interesting because in principle, this would allow us to study how you can go from one structure to another structure over something like a neutral path of change in protein sequence space. And I think that from an evolutionary perspective, this is quite interesting, but then it's still computationally difficult to study. It's purely from the size. So it's still, whenever we talk about millions of items, it becomes computationally very difficult to treat. Okay, so to finish, all of this information is available online. The methods, the results, these are all browsable, and all the code is available for reuse. And with that, I'm happy to stop here and take any questions if there are any questions.
Hi, Pedro. It looks like Sudat is having some mic and connection problems, so I'm going to step up for moderating here. We do have a couple questions for you already. The first one is, how do you cluster multi-domain proteins? If a protein has several domains with distinct 3D folds, what cluster is such protein? In what cluster is such protein?
Yeah,
Sorry, I'm reading there.
I understood the question. So again, there are multiple ways of doing these types of things. The way this was done was that it was clustered at 90% length. Therefore, in the scenario, let's say if you have a protein that has two domains, it will be clustered together with other proteins that have the same types of two domains, but it will not form a cluster with a protein that only has one of these domains. Hope that's clear. So essentially,
we have the next one. I think you can hear me and see me now, right, Gat?
Yeah.
Yeah. Next one is from Dick. Great talk. Is there any way to learn to what extent results are sensitive to AF errors, bias, i.e., a hormone sensitivity analysis?
So not for the whole set. That's computationally not tractable, or at least it would be difficult. I think Martin's lab has done a bit of analysis, but not necessarily to do with the AlphaFold sensitivity. It's more that they've done a little bit of work on parameters just to see if that would impact too much on their capacity to recover known groups of functions. There's nothing much to report on that.
Okay. The next one, it's a raised hand from Steven. Maybe I'll try to allow it. I'm not sure how it works.
Yeah. So with this one, in allowing it, Stephen, you should be able to share your mic now and ask your question out loud.
Hello, can you hear me?
Yep.
Yeah.
All right, so great. Thanks, Pedro, so first of all, great talk, nice job. I would like to thank you again for accepting our invitation to speak here today, sort of reiterate what Sudat mentioned earlier. So a couple questions, since this is really cool as we go further, exploring or expanding the protein space using AlphaFold and these prediction powers. So what about protein interactions? Some of these things, can it be possibly or reliably predict using these prediction tools to identify potential or huge number of protein interactions and how they change in different conditions? And what do you guys say there? I think there's some work I see already published from your group.
So so really what you can do is to predict the structure of the interaction using AlphaFold. We've tried this on a set about 65,000, as I briefly mentioned. And out of these, if we just directly compare with PDB, the success rate is something like 50% correct models. And you can make that more stringent by using internal confidence estimates, up to 70 to 80% correct models. But you lose many models on the way. But this presumes that you know that the two proteins share an interface. And so oftentimes what happens if you do a pull-down experiment or a yeast-2 hybrid experiment, that does not say necessarily that the proteins share an interface, just that they can interact in some way. And so the error rates for that will be higher in principle. I don't think people have really quantified that. If you want to say, can I predict interactions using AlphaFold, not the structure, but just whether they can interact. So there is signal in the data. So in fact, yes, in principle, if you take AlphaFold and just predict all by all, that would be a way of predicting interactions, but it would be a computationally costly way of predicting interactions. So it doesn't really work for that because it's just computationally too demanding. And so, as I said, we've now ran 500,000, and it's just way too slow. Basically, it's sort of, you need too much compute power. Maybe DeepMind or somebody like that could try to do that.
Cool. So one more question, if I may. So regarding the missense mutation, disease mutation that you talk about today as well, it looks like they are enriched in the pocket, as you presented earlier. My question is whether these because I heard, early days when people talk about AlphaFold, that if you have missense mutations in the protein structure, sometimes it's known to really change the protein function interactions, but AlphaFold is showing the same structure. I don't know how you comment on that, or now you have a better way to predict potential mutated protein form.
Yeah, so that's the case, and we've tried it as well ourselves. So if we have cases where a small change in the sequence is known to affect the conformation of the structure, AlphaFold does not predict that well in any way. There's been some new ideas. I'm sure you've seen some of these. There's some ideas of sort of, almost torturing AlphaFold into giving you different conformations. There's a couple of these ideas. I haven't seen nothing that sort of completely does this well. And in part is really that AlphaFold has not been trained for this purpose. It's really been trained to predict the crystallography result, right? So from a sequence. And I could speculate on why is that it doesn't work for this purpose. And so it's really more that if you have a predicted structure, then you can predict the impact of a mutation using structurally aware methods. So that's one way that we do it.
Okay, so do you have a new tool now to more accurately predict mutant protein structure?
No, I mean, we use actually very old approaches, empirical force fields. This is, I think, Creston's lab has built a neural network model that tries to replace this. And we were thinking about similar things. In the end, the neural network at best will learn the empirical potentials. And so it will be faster to use. Some of these things are quite slow. But I don't think we have enough training data to come up with something that will be better than these established old-fashioned empirical potentials. I'm not sure, but it's worth trying for sure.
All right, cool. Yeah, all right. I think I will follow up later by email with more questions. Thank you.
Yeah, thank you.
Thank you, Stephen. We have one more question from Hiram. How did the clustering account for proteins with large amounts of disorder?
Yep. So these tend to be not clustered into the clusters, essentially. So the current structural alphabet penalizes against them. So they're usually not going into the clusters. And of course, this is a big part of it, right? There's a lot of unstructured proteins that maybe fold upon binding or, fold in specific types of conditions, or simply they don't have structure. And then the ensembles is the function. That's, there's many thoughts about that. And of course, this does not take into account that type of, let's call it, structure.
I do have one question for myself. You say that you found a lot of species-specific cluster. Are they coming from some weird organism that nobody actually studies, or what organism they are usually from?
No, they're all over. So human has 13 of these. I mean, then we looked in detail for the human ones, and they were not convincing most of the time. I think there was one that maybe looked like something that was convincing, as in it seems not homologous to anything, but it looks like a real protein. But no, they're all over the place. There is these clusters of things that don't match other proteins in other species.
Okay, I can see another question from Alexi. Is there any way to account for effects on 3D by PTMs?
Yeah, we wish. So we work on phosphorylation quite a lot. So that was one of the examples that we tried to predict the impact of phosphorylation on structures. It does not work. Yeah, so maybe they'll come up. Again, I think a lot of it has to do with the fact that AlphaFold predicts one conformation, and proteins exist in multiple conformations. And maybe the information is somewhere in the model, but so far people haven't very successfully being able to do this. Again, so there are different ways of trying to generate ensembles now, or different conformations at least. And so maybe there are some good ways of trying to do this. For phosphorylation, it's still possible because we can essentially mutate it to a negative charge amino acid, and that will mimic the phosphorylation event. For other PTMs, this will be much harder.